I am Nurul Fatiha with the matrix number D19A0020 will bring you on a journey to discover Pasteurellosis in fish. Pasteurellosis is caused by a bacteria called as Photobacterium Damselae subspecies Piscicida. This is a part of the thermotolerant group of the genus Photobacterium. These cells can enter a viable but not culturable state in response to environmental stresses such as starvation, antibiotic exposure, or lower temperature that allows the cell to enter a state of dormancy and survive until conditions allow resuscitation and reinitiation of infection, which will then contribute to the survival of virulent strains. This disease can occur in a variety of marine fishes, including Gillhead sea bream, red porgy, red sea bream, European sea bass, meager, and sole. This disease is distributed in Japan, Taiwan, China, Portugal, and the Mediterranean. The clinical signs of pasteurellosis depends on the phase. For hyperacute or acute septicemia that occurs in the hatcheries, infected fishes often found dead in large numbers on the bottom of the tank with only a few darker fishes swimming sluggishly and off balance near the surface, often showing nervous convulsions prior to death. For acute pasteurellosis, there are no alarming signs with fish behaving and feeding normally before the disease strikes, causing many dead fish to carry amounts of feed in their stomach and gut. Convulsive erratic swimming before death often comprises the only clinical signs of acute outbreaks. Anorexia, lethargy, darkening, and ulceration of the skin follow shortly afterwards. For subacute pasteurellosis, gills become pale with excessive mucus secretions, with congested, inflamed patches, and often focal necrosis next to congested areas. Lip, orbicular skin, and lower jaw inflammation and necrotic skin patches on the body flanks, dorsal area, and tail become common. The fins, mainly pelvic, dorsal, and caudal, may be eroded. Skin and fin erosions are covered with mucus, thus the lesions appear in the water as white patches. For chronic pasteurellosis, the signs may not be as apparent as the acute form. This bacteria spread via infected phagocytes, mainly macrophages, and the spread is rapid, with lethal effects after a few days of infection. Ion acquisition from its host by means of ion-binding siderophores increase virulence, and the cytotoxic extracellular products damage the infected cells with the consequent release of the bacterium and the invasion of adjacent cells. Pasteurellosis is a temperature-dependent septicemic disease. Outbreaks occur when the water temperature rises above 18 degrees Celsius. In hatcheries, where warm borehole water is in use, pasteurellosis is a major threat all year round. Pasteurellosis can be diagnosed through postmortem. For the acute phase, internal lesions are often absent at necropsy. For the subacute phase, the liver is most often inflamed and congested. The spleen is enlarged. The kidney is pale and edematous. The intestine carries a moderate quantity of fluid and some whitish mucus clots. The swim bladder is not distended, thus the majority of dead fish have sunk to the bottom of the tank. For the chronic phase, typical pseudotuberculi develop mainly in the spleen and/or in the kidney parenchyma. They comprise creamy white granulomatous nodules composed of masses of bacterial cells, epithelial cells, fibroblasts, phagocytes, and necrotic cell debris. These have led to the descriptive name of pseudotuberculosis. Bacteria accumulate in the phagocytes, capillaries, and interstitial spaces, and bacteremia is pronounced. Sampling is done for diagnosis. Freshly dead fish target organs such as spleen, liver, and kidney. Fish eggs, live prey organisms, or even tank and filter sediments may be submitted for PCR examination. For the treatment of pasteurellosis, it is hard to eradicate by antibiotic treatments due to the intracellular location of the bacterium, which seems to protect from circulating antibodies after vaccination against the disease and may explain the low RPS conferred by the vaccination, as well as the short duration of immunity post-vaccination. transferable genetic elements or are plasmids carrying the genes of resistance to many antibiotics. Carriers may persist on farms and may show disease under stressful conditions. The only commercially available vaccine is an ECP-enriched bacterium preparation that has been employed in several European countries. The recommended vaccination protocol consists of 2 bath immersions at monthly intervals, starting at the larval stage, when the fish is 50 milligrams, and an oral booster immunization when the fish reaches 2 grams in body weight. That is all for Pasteurellosis in fish. Thank you.